So could you tell me the results of your recent study?
Sure. So let me do a quick bridge between the parabiosis, right? So the parabiosis is like a Siamese twinning of the young and the old animal. And they share blood, but they also, because they're connected for a very long time, a relatively long time, they share the tissue and organ systems of the partner. So the young animal suffers half its mass or more than half its mass being old and having old liver, old kidneys, blah, blah, blah. The old partner gets access to a young set of liver and kidneys and pancreas and insulin regulation and digestive system and whatever. So we didn't know for sure if it was the effects that we saw were because of the blood or because of the other systems that kind of communicate through the blood. The next experiment we did was a blood exchange. So we just took two animals and transfused blood back and forth between them. And then after several exchanges, you approach homogeneity between them. So there's about 50-50, and then disconnect them. And because the only thing that was transferred was the blood, we could rule out the effects of organs and regulatory systems. So from that work, we found that the liver was improved in the old animal, the muscle regeneration was improved, the brain wasn't really all that improved. And the young partner that got old blood, the muscle regeneration was a little worse. The liver couldn't really see much of a defect for the things that we looked at, but their brains looked like they got stupider. So, so then at the end of that, we said, okay, now did this happen because the old animal got an infusion of young factors, young pro-youth factors, or did it see improvement because it got rid of half of its old bad factors? And conversely, the young animal, did it get worse because it got these old factors or because it got rid of half of its good factors? So what if we could do a blood exchange like we did between the young and the old animals, but with each of them, have them exchange with something that was neutral. And we tried to figure out what age of mouse could be neutral for, but it's difficult to point to a particular age. So we said, well, what if we just hypothesize, say, okay, let's say it's not the red blood cells and it's not the white blood cells, and that leaves plasma. So what if we had some blood that we throw out the plasma and replace it with purified saline and purified albumin, which is like the major ingredient in plasma, but it doesn't have any factors in it. It's just devoid of factors. It doesn't have any old factors, doesn't have any young factors. And do the exchange with that, would the old animal look a little younger and would the young animal look a little older? So that was the basis for the experiment that we did. So we took old mouse blood and pelleted the red blood cells and the white blood cells and threw away the platelet-rich plasma, right? So it's got albumin in there, it's got antibodies in there, it's got platelets for clotting, and it's got all the old factors that would be in old blood. And replace that with saline, mixed in with the major purified component of blood, is albumin. And then did the exchange with the old mouse with that, and we did some with young mice too, so that in the end, the old animal has all its red blood cells and has all its white blood cells, and has half of its platelets, and has its plasma diluted in half by this purified plasma, basically. So that was technically what the experiment is. And then what we found was surprising. I didn't expect the old animal to be really all that much improved. I figured some things might change, but in other ways it'd get worse. But it didn't seem to get worse in pretty much everything we looked at.